My name is Ruth. I am not Brad Schaefer. Brad is stuck in traffic somewhere between here and the South Bay. He called me about 10 minutes ago and said, hey, can you do the introduction from Butch? I'm not going to make it. For those of you who know Brad, you can hear him say this. Butch even said, oh yeah, I thought it was last week. He was Anyway, it's my honor on behalf of Brad and the Major Issues in Modern Biology series endowed by the Storr Lecture Endowment, which I'll talk about in just a second, to introduce today's speaker in the Major Issues in Modern Biology series, Butch Brodie. Before I do, I think it's important to realize that you too, if you want to leave an enduring legacy to UC Davis, can do something like the Storrs did. And in the center of this photograph is a picture of Tracy and Ruth Storr, Tracy on the right, Ruth on the left, in case you didn't know that. And both of them left some of the most enduring legacies to Yolo County and the University of California that just about anybody can leave. Ruth was the first pediatrician and the first woman doctor in the county. And she opened up throughout the county a whole series of children's clinics whose legacy continues to endure today in some of the best child care facilities in the state of California. She was also an avid gardener, and many of you still enjoy the legacy that she and her donations have left in the Ruth Storr garden down in the University Arboretum. For many of us in ecology and evolution, we live in Storr Hall, and Storr Hall was named after Tracy Storr. It was built in 1969, the year after he retired. He was the founder of the Department of Zoology at UC Davis. This was in 1923, and he was, for almost 40 years, the only member of the Department of Zoology. Tracy accumulated money, and there are many stories about how he accumulated that money, but he and Ruth managed to accumulate enough money to endow a series of scholarships and lecture series and a whole variety of foundations throughout the academic world and the arts world, including trying to remember what it is in the Society of Icks and Herps, but there's
a student paper.
A student paper prize in the Society of Icks and Herps. Many of you don't know that many of you know that Tracy Storr was one of the great natural historians in the state of California. He wrote the textbook General Zoology in 1949. That, although it was, I just assure you, it was written before I was born, and it was not the zoology textbook I used. Many generations of zoologists learned their zoology from Tracy. And for those of us who love the Sierra Nevada, you know that Tracy Storr literally wrote the book on the natural history of the Sierra Nevada. What some of you don't know is that Tracy Storr is responsible for the modern design of the California state flag. And in fact, this is what the grizzly bear looked like on the original California state flag, and it's been described as a hybrid between a dog, a pig, and a bear. And Tracy, as it turned out, was one of the few living people ever to have seen a living California grizzly bear. And so he did the drawing of what is now the California grizzly bear on the state flag. Good little bit of trivia, huh? You should also know that today's speaker follows in the footsteps of Tracy Storr. Tracy was also an eminent herpetologist. Brad reminded me of that. And so is Butch Brodie, today's speaker. And Butch follows in some interesting footsteps himself. Butch is not really in this, well, Edmund Butch Brodie III is not in this picture, but Edmund Brodie Jr. is in this picture, Butch's dad. And you'll see Butch is not aged at all. Some of you may or may not recognize other colleagues of yours in this picture. That's Carol Hom. Wow. And how many of you people can see where Steve Arnold is? Carol knows where Steve Arnold is because she provided me with this picture. There's Steve right there. Wow.
With a cigarette.
With a cigarette. So, like his dad, Butchie is first and foremost, and like Tracy Storr, a herpetologist. He's also considered, and in my view, one of the really great natural historians. And when students, and when I was president of the American Society of Naturalists, I gave a talk on the new natural history, and people asked me, isn't natural history dead? And I said, nah, there are people like Butch Brodie who are doing the kind of natural history that epitomizes what the state of the art should be at the interface between ecology and evolutionary biology in the 21st century. Butch is an eminent quantitative geneticist, behavioral ecologist, particularly well known for his studies of predator-prey interactions and sexual selection. He's a phylogeographer, he's an ecological geneticist, he's a chemical ecologist, as you'll see, and he's well known for his studies on coevolution between predators and their prey and the role of chemicals in mediating, chemical evolution in mediating those interactions. He's had countless honors, he's published hundreds of papers, and I urge you all to enjoy your last meal if you ate one of these, and hope that there wasn't teratatoxin in the snakes that you guys ate. Butch is the only speaker I've known in 26 years here at UC Davis to be honored with the likeness of his favorite organism in a cookie. And so it's a pleasure to introduce Butch Brodie, who will be talking about time to change the channel. I can't quite remember the rest of it.
Thank you. That was outstanding. I'm not going to call the picture back up, but I want to point out that Steve Arnold, who's my academic father, might have looked a little different, but he's wearing exactly the same shirt and overshirt that he still wears today. And I don't mean like the same color. I mean it's the same shirt. All right, this is the greatest thing. Whoever's responsible for this, I've already emailed a picture to my kids and my wife, because I think that's...
But some of those will make you very, very sad.
Well, let's see if we can figure out which ones they are by the end of the talk. All right. I'm going to do just a couple-minute commercial. You guys have here at close hand more beautiful places to work. I've worked probably more in this area, including Oregon, Washington, California, than I have out east. But my current gig is in part as director of a field station, and it's my responsibility to tell people what's there and that it's there for you. If you ever need a place on the East Coast in the Appalachians in deciduous hardwood forest at the top of a beautiful mountain to work in the summer, come and visit. If you are looking for a place to send your independent undergraduates who are spectacular and want to do an internship, we run an REU project. And we do summer courses, and we're on the way to doing some workshops for graduate students. So I'm not going to belabor any of this. I just want to show you some pictures to tease you and point out that we're about to announce early career fellowships. So these are essentially cost-free opportunities to come and use the station to develop new projects. So these are for senior students, postdocs, new faculty. The idea is that if you come and you see what a field station has to offer, you will like to stay there. And so I'm saying this general, I'm saying it specifically about Mountain Lake, but really use your local field stations. These are spectacular places that are communities of biogeeks looking for other biogeeks to play with. These are what we have on the East Coast. These are a different set of salamanders than you're probably used to. I'm not going to talk about the courses so much, but these are the workshops that we're starting. It looks like this year's workshop, these are 10-day to 2-week workshops at the graduate level, will probably be an Eco-Evo-Devo workshop led by, I can't name the person yet, but one of the leaders in Evo-Devo genetic approaches using the ecological format of a field station to show you guys how we can take those techniques into the field. We're really excited about this. So the advantage of this is it's not a UVA credit, which means you don't pay a UVA tuition. So these are really experiences for grad students to get expertise in a very specialized area that we're starting this year. And then the REU program, most of you know about REU programs. Tell your undergrads about it. These are the greatest things going. We're approaching our 20th year for an REU program at Mountain Lake. The coolest thing that we do every year is invite back the first and second and third generation REU people who are now teaching classes and giving the named lectures at the station in the summer. It's really an unbelievable vault into a career to do an REU program at a station, and we're one of many. So if you're an undergrad or if you have undergrads in the lab, send them to the NSF website to look at those lists of options and get them out there. They're competitive. but they are just the greatest experience they're ever going to have. All right, so that's the commercial. This is the full title that neither one of us could remember. Predator-prey arms races and the evolution of toxin resistance in snakes. It is a family affair in a lot of ways. My collaborator is my dad, who it turns out looks a lot like me for some reason. As a quantitative geneticist, that's probably shouldn't be so much of a surprise. And the drawing is by my wife, who's an artist. And this is really all you need to know, and we could stop at the end of this. Snakes eat newts, and there's a whole lot of interesting biology that proceeds out of this. So I'm going to talk about the natural history, not surprisingly, after the introduction, to kind of set the stage and talk about what coevolution is about. To me, it's really about the interaction of traits, more so than species. So a lot of times you'll hear people talk about coevolution between species A and species B. I think about it as the evolution of one trait against or with another trait. They happen to be in different species. All right, so to me, the emphasis is on the phenotypic interface, or the traits that are mediating the ecological interaction that drive coevolution. I'll talk about some of the earlier work we did on the geographic structure and the mosaic of evolution. So how does it change from space to space in a landscape? And what does that tell us about how coevolution proceeds, if anything? And does it tell us anything about the general dynamic by being able to survey the interaction over many different localities? And then the last part of the talk is work that we're working on currently, which is really geared at getting more mechanistic, which is a place I never thought I would get to as an ecologist first, natural historian, and evolutionary biologist. But really getting down to the mechanism of how these traits are made has opened some doors to understanding the way that, or the explanations we think for the patterns that we see. So they address several general issues in the genetic basis of adaptation, but I think more so for us are really showing us why we see the patterns in nature that we do. All right, so page 1, everybody in the same place. Predator-prey interactions are almost invariably talked about as the arms race. So this goes back to the coyote and the roadrunner and all of these cartoonish versions of the world. And the bottom line is there's very little evidence that this really ever happens. So what we usually see are really elaborate adaptations of prey to specific predators, but not the other way around. We don't see a lot of evidence of predators that have evolved specific characteristics to exploit or defend themselves against a specific prey species. So empirically, there's not a lot of support. The second part is a conceptual one, and it has to do with the asymmetry of selection that's imagined to work. So Dawkins and Krebs called this the life dinner principle. And in a nutshell, a vine snake like this Leptophis has less to lose in an interaction with an individual prey item, the Norops, or Anolis, depending on your systematic inclinations, than the lizard does, right? The lizard is losing its life, fitness goes to 0 immediately. The snake loses the interaction, it loses a meal, and it lives to try again another day. So this, in the mind of Krebs and Dawkins in this early arms race paper, meant that selection was much stronger on prey than predators, and so we would expect to see predators left behind in a race and prey to get ahead, and that would erase the arms race. The other way to think about this asymmetry is really in the consequence, what we think of as dodging the bullet. So this is a horned lizard that was caught by a shrike. It's very unfortunate. It's stuck on a stick. Shrike peels the meat off the body. And obviously, the fitness of the horned lizard went to 0. Right? The shrike may have severe consequences if it loses a meal, or it might have no consequence at all. And this is going to depend on things like whether there are other available prey, whether it's feeding young at the nest, whether it's had a meal in the last few days. So the variability in that fitness consequence may be much higher for a predator than it is for a prey. And that again affects the regression, which is the way we think about selection. So selection in either one of these scenarios could be much weaker on predators than it is on prey. The exception to those scenarios is when we get dangerous prey. Things like poisonous prey, things with big nasty teeth or horns that could kill the predator when they attack the prey item. A really great example of this I learned about a couple weeks ago at Minnesota's work that Sharon Jansa has done on opossums that eat venomous snakes in Central and South America. And they've evolved blood proteins. It's actually the von Willebrand factor. It's a clotting protein. The lineages that eat venomous snakes have evolved resistance to that venom. They're not prey on the snakes. They are preying upon the snakes. So this is another example of predators dealing with dangerous prey that seem to have evolved a specific adaptation in response to that danger. Right. So that's the setup. What we're really talking about is not predators or prey, but a molecule that sits at this phenotypic interface. And this is tetrodotoxin, or TTX. I'm not a biochemist. I can't really tell you much about the molecule, except that it works like a cork in sodium channels. So sodium channels are critical for all nerve and muscle function. They are responsible for the electric current that moves along a membrane. This is a cork that stops the transmission of the positive charge in and out of that membrane. And basically, without that function, you die. The immediate cause of death is usually asphyxiation. Right? Anything with sodium channels is affected. So this is essentially all vertebrates. Virtually all invertebrates have TTX-sensitive channels. So amazingly broad toxicity. Not the topic of today's talk, but one of the interesting aspects of the toxin is the identical molecules found in at least 6 different phyla. Right? So we'll talk about newts. It was first identified in puffers. It's known in the salivary glands of blue-ringed octopus. There's a starfish and a crab and a couple of marine snails and now a terrestrial flatworm that have been identified with TTX. Same molecule, right? Nobody's really sure where it comes from. When you see things like that, everybody assumes it's symbiotic bacteria. I think there's some evidence for that in the marine organisms. There's not very good evidence in the newts. And we're not going to go much further with talking about the newt side today. So this is one of those things where if you were really doing good coevolutionary work, we'd have been working on the newt side as hard as we work on the snake side. And I have to admit that we haven't done that. So it's in a vessel. TTX is in the wild in something that's probably familiar to most of you guys. When I give a talk, I have to explain all about Tarica, the rough-skinned newt. If you guys have been out on a rainy day anywhere in the Coast Range or the Sierra, you've probably run into Tarica. And they look like this most of the time, about 6 inches long, fairly innocuous. You walk up to them, they do nothing. If you're a bird and you peck them, or if you're a herpetologist and you do what herpetologists do, which is grab things, then they invariably go into this thing called an Unken reflex, which is advertising an aposematic signal on the belly, the orange venter, advertising chemical toxicity, basically telling predators that it has a lot of tetrodotoxin. What's interesting about Tarica, different than its sister genus in the east, Notophthalmus, and other newts, is that it has a ridiculous amount of toxin. So depending on how you want to do this math, enough to kill 20 people by mass, right? Probably not my people, but some other people. A lot of mammal tissue. So that's based on a mouse toxicity unit. I like to let my collaborator explain it because he does it more colorfully.
This is probably the most poisonous animal in the world, with enough skin toxin to kill tens of thousands of mice or perhaps 100 people. Of course, they don't bite, so this isn't really dangerous holding it unless I were to eat it, which I won't.
I'll give it to my son.
I never get tired of doing that to him. He says 100. You know, this is all based on the reaction of a mouse sodium channel to the toxin, and based on the old school methods of looking at toxicity, which are LD50s. You know, you essentially take a sample and you iteratively hit that sample of subjects with an increasing dose of toxin until you find the dose that kills 50% of your subjects. So besides the ethical issues of how many mouse souls you might incur there, it's not a very good measure for looking at individual variation in toxicity or resistance. Right? So we don't do that. I'll explain how we assay the toxicity in a bit. We wouldn't have much story if we didn't have a predator that was resistant. So these are all West Coast Thamnophis sertalis. I won't even show you the East Coast version of it because it's dull and brown and not that exciting. The one that is most similar to the local snakes is here. This one's from Gilroy, California. This is Willamette Valley of Oregon. What you need to know to start the story is that everywhere they occur with newts, they eat newts. But nowhere is this the kind of specialized interaction that we usually think about with, say, plants and herbivores. So where we've been able to sample the diet, 1% is about the maximum percentage in the diet, right? So it's really a rare prey item, and that leaves a very strange ecological puzzle here. There are geographic differences in how much resistance there is in these snake populations, and I'll belabor that, so I won't say more about it. There are costs, at least there appear to be costs in terms of locomotor performance. So if you race a snake on a racetrack, the fastest snakes are the ones likely to be the least resistant to the toxin. The ones that are most resistant to the toxin are slow. That's also true at the family level, so there's a genetic basis to that trade-off. So there seems to be reduced locomotor performance that correlates with the ability to resist the toxin, right? And then this, let me spend a minute on this one. It's always been a puzzle how you get the evolution of a deadly toxin or resistance to a deadly toxin. If you kill your predator in the process of deterring it, then there's no learning opportunity. There's no variance in fitness. If the predator dies when it encounters any prey item, there's no selection for quantitative differences in the toxin. So once you have a deadly toxin and resistance to it, it's very hard to imagine the continuous form of selection that drives continued arms races or escalation. Okay? So this was an experiment done by Becky Williams, who was a grad student in my dad's lab, and she interacted newts and snakes. Newts from a more resistant, more toxic population than the snakes could really handle. And what she found was that not all snakes finish the ingestion phase. So they would attack the newt, they would begin to swallow, and then between 5 minutes and 55 minutes, so an hour, the snakes would yawn and kind of, if you've ever watched a snake eat, they kind of do it in reverse. They back their mouth off of the newt, and the newt pushes away and walks off. So an hour with its head and shoulders in the esophagus of a snake, a newt can walk away apparently unscathed, right? What predicts whether that interaction goes all the way to ingestion is the individual level of toxicity of the newts and the individual level of resistance of the snakes. So there's the quantitative relationship that's necessary to drive the whole dynamic that we're talking about. And it was years before we figured out that there was that sort of behavioral self-assessment. And I think it's not, measuring the toxicity. I think it's literally the snake experiencing an intoxication with too much toxin, right? And then I'll get to this at the end. We now know that there are other species of garter snakes and maybe other species of snakes worldwide that have evolved resistance, okay? So this is what we do. This is, it's not rocket science. You jump on snakes. One of the neat things about snakes is that they are all carnivorous and they all eat their prey whole. So they're ideal for ecological diet studies. You can do this procedure, which is known in the business as barfing the snake. And if you're lucky and you've found one that's recently eaten a prey item, you can coax the prey item out of the belly. This always grosses people out. I went to a med school once, and I followed a guy who gave a talk on some kind of cancerous tumors. It had all these nasty slides of things cut out of human bodies. Nobody made a noise. I showed that clip, and everyone just, ugh. So this is an adult newt. It's probably been in the belly of that snake for about 6 hours, based on the digestedness of it. So that's a rare event, and that was a really lucky thing to catch on film. This is from one of the Oregon populations that's really resistant. So based on that sort of information, that's how we know that nowhere do they eat a lot of newts, but everywhere they co-occur, at least occasionally, they're picking a newt up, right? So the story is, that's the natural history background. The story is that we're trying to understand the evolution of resistance. And as I mentioned, the LD50 is not an ideal way to get individual measures. So to measure variation, you really have to get individual, family level measures that you can then look at resemblance within families, resemblance between families. We had to come up with a new approach because we couldn't kill that many snakes that we would need to do to calculate an LD50. So we came up with this bioassay where we race snakes on a racetrack, figure out how fast an individual goes. So this is a baby snake moving down a track with infrared sensors that's hooked up to a computer. It trips the beam and it tells you how fast the snake crawls. You wait a couple of days, you inject it with a known dose of tetrodotoxin, and then wait 30 minutes and race it again. And so now you can look at the performance deficit as a continuous measure of how resistant to the effects of the toxin it is. When snakes can't crawl, they resort to other kinds of defensive behaviors. Baby garter snakes are not very scary. When they do finally crawl, they're very slow. Okay, so that performance deficit is what we're going to use now as our measure of resistance. So when we talk about resistance, what we're really talking about is this difference between the baseline and the post-injection speed, essentially the percentage. So 100% on the graphs that you'll see in the rest of the talk is unaffected. They crawl as fast as they did pre-injection. 0% means totally immobilized. And this is a classic dose-response curve that looks like this. So there are asymptotes, and really we think we've got accuracy between around 15% and 85%. Beyond that, we're saturating. All right? It's a really nice approach because you don't kill the animal, you can retest individuals. It's highly repeatable. It is ecologically relevant in the sense that snakes, the first effect of intoxication on a snake is immobility. So if we had known that that snake in the video had eaten a newt, I would not have had to dive onto the stupid thing. Because you can literally go up and toe poke it and it won't move. Right? There's that much effect of the toxin early on. Right? The other advantage is that the level of toxicity that we can inject them with is non-lethal. So we can adjust that amount and then look at variant structure within populations that haven't yet evolved a trait that would be ecologically important. So if you were to feed a newt to a snake, you'd kill it. But we can use low doses and look at that variant structure, and that becomes very important for us. Right? So we've used this approach to ask a lot of questions. Again, I'm going to talk about the geographic structure and then the genetic basis. A lot of our earlier work was focused on John Thompson's ideas about geographic mosaics, which to me are less hypotheses and more perspective. You know, as a population geneticist, you kind of appreciate the idea that evolution proceeds at the population level, and ecologically the interactions may vary from one population to another. And the geographic mosaic in its simple form just notices that interactions might vary in space, and when that happens, coevolution varies in space. Right? So the first prediction of that is that we should see places where coevolution is intense and places where coevolution is non-existent, hot spots and cold spots. And then from that, you see these other predictions, like the maintenance of genetic variation, where what Thompson's called trait remixing, I think is mostly gene flow, from hot spots and cold spots come together. So we might see more genetic variation in some places than we would expect if we were seeing strong selection weeding out genetic variation. That should lead to places in a geographic landscape where we get maladapted traits. And I don't like the word, but essentially mismatched abilities. So places where predators have a higher phenotypic level than they need to deal with the predator. Predators have a higher phenotypic ability than they need to deal with the prey, or prey are more defended than they need to deal with the local predator. Right? And if this is all true, then if we played this tape out in multiple lineages or in multiple parts of a geographic range of interaction, then we should see different evolutionary outcomes, different results to that arms race. Okay? So what do we get out of snakes and newts when we look at this? Just as a reminder, this is what we're doing. We're racing snakes, injecting them, racing them again. And these are the data, sort of characteristic dose-response data from 5 different populations. So you'll see on a log scale the mass-adjusted mouse unit. This is baggage of working with your father. He invented a unit years ago, and we had to use it ever since. So this is essentially the milligrams of TTX per gram of snake adjusted to how much it takes to kill a mouse, a 10-gram mouse. So just think of it as a mass-adjusted dosage. Each of these lines is a regression based on data from a single population, and we interpolate a 50% dose. So this is the performance dose that knocks down the average snake to 50% of its crawl speed. So It's not a matching level, but this is one with the most variable outcomes to interactions between a given level of toxicity and the range of variation of resistance in newts. So these are 5 statistically differentiable levels of resistance. Every one of these colors on the graphs you'll see is a statistically defensible level of resistance. Purple and blue are essentially non-resistant in the terms of a West Coast newt. These are not resistant enough to eat a Tarica and survive. Green and up is some level of resistance. Right? So again, examples. Here's what we get when we look across most of California, Pacific Northwest. Huge range of variation, 3 orders of magnitude in level of resistance. You've got populations up here where it takes 1,000 mammoos to knock down a snake to half of its crawl speed. We literally have populations that we cannot afford enough tetrodotoxin to test, that's really resistant. It's an interesting way to think about it. I hadn't really thought about it. It's $300 a milligram, almost 400 now. Anyway, all of these populations in purple and blue are sort of baseline level for that clade, natracine snakes. Right? So this doesn't tell us much about the overall resistance level, the overall geographic pattern. If you put it on a map, most of you guys are used to looking at West Coast maps. The gray area is the range of the genus Tarica. These are all Thamnophis sertalis populations. The purple spots out here tend to fall out, as you might guess, in zones of allopatry. So outside the range of newts, this was the first part of what we were trying to figure out. Outside the range of newts, we don't see the evolution of resistance. That's surely not a surprise, but that was sort of the original motivation. We thought we would see 2 colors, non-resistant, super resistant wherever the gray in this distribution was. You've now had enough time to realize that that's not the pattern. Right? We see everything from non-resistant snakes up here in Northern California and British Columbia to highly resistant snakes in Oregon and parts of California. Just this huge range of variation that's not at all what we expected. And you can boil it down and do a little bit of smoothing to it to realize that you've got one apparent hotspot of resistance in the Bay Area and the adjacent Sierra, and another one up here around the Willamette Valley, Corvallis area, and the adjacent coast. So if you put the smoothing on that, it looks like a nice clinal radiating pattern of resistance, right? Where you've got hot spots and movement of those genes for resistance out from those hot spots. Now, there are other explanations. It's possible that selection is variable throughout this range, and rather than resistance evolving here and moving genes out, that this is actually matching all along the way. And that's one of the things we'll look at. Right? So this was the pattern. The other piece of the puzzle that I don't have a slide for this time is polymorphism. So one of the predictions is we should see polymorphism. We haven't sampled really broadly, but if you come up here to the Russian River, mouth of the Russian River in Sonoma County, or down here in the Monterey area, there's pretty good evidence that we're seeing multiple alleles for resistance, and I'll show you some of those data later. So there seem to be places where lots of variation is being maintained. All right. So the first question, of course, is why do we get 2 hotspots? Is this coevolution in 2 different places and independent origins, or is this historical artifact, one origin of resistance in the ancestor of those 2 lineages that then somehow invaded those 2 places separately? And really simply, that boils down to looking at the phylogeographic pattern. We recruited Fred Jansen's lab at Iowa State to help us out with this. So he did a mitochondrial phylogeography of this area. It's not well resolved. Probably not a surprise given the time we've had for invasion here. We get one clade down here that corresponds to most of the California populations, one here that's sort of Intermountain West and Northern Canada, and one here that is the Coast Range, Northern Coast Range. Right? So when you map the traits onto that, you get a clear pattern of 2 origins of resistance. And they're clear not because of the phylogeny, but because the ancestral condition is rooted really well. We've got a lot of data from other populations. So we've got at least an origin here in the Oregon populations, and 1 or possibly 2 in California. We know now that this Sierra population, there's some pretty good support that that's not closely related to the Bay Area populations. Although, as Brad and I talked about last night, snake phylogenies and phylogeography are pretty poor right now, so we still don't have a great picture there for California. So how do those match up to the presumptive selection pressure, the newt toxicity level? So these are HPLC plots representative from two newts, one from the hotspot in Oregon, one from British Columbia, Vancouver Island. HPLC, you run a reaction, you look at products at a particular time, and you can identify the product by its time, and the area under the curve tells you how much is there. Here's TTX for the Oregon newts. There are no peaks at all for Vancouver Island. So this was shock number one. We thought all newts were, I guess it's shock number two. We thought all newts were toxic. If you match those up, you get a dandy line. Non-resistant, no toxicity. Resistant, high toxicity. Smart scientist stops when you get a regression with an R squared of 1. A good scientist keeps going. So we pursued it. Now this is work that Charles Hannafin did, where he took all the snake populations, went back and sampled newts and asked what were the levels of toxicity. Essentially, do we see the matching pattern that you would expect with, with an arms race interaction? Or do we see places where they're out of sync functionally? So now the plot's a little bit different. This is average snake resistance, whole animal level. This is average newt toxicity, whole animal level of toxicity. Each one of these dots corresponds to one of these populations. They're colored by newt toxicity level. This solid line is the 50% level of resistance. So how much toxin does it take to take an average snake down to 50% of its crawl speed? Above that is the 15% dose, below 85. So what that means is that below that lower bound, we've got a phenotypic interaction zone where snakes are unaffected by that level of toxicity. A snake of that resistance, anything in here, is so resistant that interacting with a newt in that zone doesn't have any fitness consequence. It's not slowed down at all. Presumably, there's no opportunity for reciprocal selection. Above that zone, we have the converse, where there's so much toxin in the prey that they would kill any of the potential predators there. Right? And you can immediately see we don't get any localities up here. We only see localities down here. Right? In between is something we call loosely matching, and we don't really know that this is functionally matched. We just know that there's an opportunity for reciprocal selection there because you might get variable fitness outcomes. Some snakes lose an interaction, some newts lose an interaction. Right? The matched populations occur throughout the range. Right? Virtually everywhere there's an example of a matched population. There are really two kinds of unmatched populations. There are localities. There are those here with low levels of toxicity and resistance, and those with high levels of toxicity and resistance. Right? The interesting ones to us, or the most interesting ones, are these that correspond to essentially Sonoma County, the San Francisco Bay garter snake. So we had some zoo litters that were a little bit inbred, but probably still representative. I think that's Gilroy. No, that's East Bay, Berkeley area, and then one over in the nearby Sierra. All of these populations, localities, have newts that are highly toxic and snakes that are highly resistant, but so resistant that the local newts can't hurt them, essentially. Right? So how did they get out? You know, we think about this, we quip about it as winning the arms race. It's a lousy metaphor, but they've somehow phenotypically moved from this zone of reciprocal selection out into a zone of no reciprocal selection. Right? So we see a lot of evidence for the mismatch. We see some that are so far out that there's really no opportunity for reciprocal selection, what we would think of as a cold spot. And we see others, about 50%, where there's some level of matching where we might expect variable fitness outcomes. I didn't say much about this. I'll come back to it in a second. All right. So now we go back to the original sort of Klein maps of resistance and toxicity. This is the way we were analyzing things a few years back, where we were looking at snake resistance on a geographic scale, newt toxicity on a geographic scale. You do the vision test, and they match up really nicely. places where you have high levels of toxicity, you have high levels of resistance. Down here in California is the same thing. So what Charles did was develop essentially a mismatch measure and ask, if you do that functional analysis in that last plot, how do they line up? Well, it turns out that the places with the most extreme resistance and toxicity here in California are the most mismatched. So functionally, they're not close to one another. Places up here in Mendocino where you've got really relatively low levels of toxicity and resistance, they're closely matched. There's probably high potential for reciprocal selection. Likewise here, and then through most of the Olympic Peninsula in northern Oregon. So there's no consistency with the phenotypic exaggeration and what we would imagine is the strength of coevolution. And that was a real shock to us and made us eat a little crow on our original hotspot analysis. Right? So what do we think is going on? This is the arm waving slide. Right? So this is the pattern of matching within populations. These turn out to be pretty close to the ancestral reconstructions for a resistance level in snakes and toxicity levels in the clade of Salamandrini, the newts that are mostly Asian and European with 2 genera in the US. Right? So we think things start as an ancestral condition for both taxa, and then something causes newts to increase their toxicity. We have some ideas, but really nothing concrete yet. A guy named Brian Gall, who's in my dad's lab at Utah State, has been working on caddisfly predation on eggs, and it looks like that may be the key axis, and I can tell you more about that if you're interested. Then we get into the zone of rough matching and reciprocal selection, things that where we get variable outcomes, and then something happens that launches one of the species out of the arms race, essentially escape in phenotypic space. We only see it one direction. We only ever see predators essentially escaping from prey and never the other way where prey escape. And that's exactly the opposite prediction of that life dinner principle that I laid out in the beginning. We would expect, based on those arguments, that prey should escape rather than predators, and that's not at all what we see. And that may be a special case of dealing with toxic prey. Right? I should have, oh shoot. We have to go through all this again. I should mention, just to reinforce what an arm waving scenario that is, I showed this to Kurt Lively one time and gave him the story, and he said, yeah, well, that's just like a red queen because costs are now going to drag resistance back here, and you're going to get a nice cycle. Right? So completely consistent, except that we never see any of this intermediate space occupied so far. So where we are now is looking at the mechanisms and trying to understand whether we can explain that jump out of the arms race based on some understanding of the genetics of the traits, and whether we can figure out something general about the genetics of adaptation from this system. So the first is asking, what is the mechanism? Turns out to be sodium channels. Where does the source for this adaptation come from? This is essentially the argument about new mutation versus standing variation. It's much faster and easier to evolve adaptation out of standing variation in a population than it is new mutation, so you might expect that to be the result. On the other hand, that requires that you've got neutral or nearly neutral mutations floating around it, segregating at some frequency. How convergent do we see, or how much convergence do we see across deep clades? If this is a common selection pattern, we might expect to see other species in other parts of the world that have experienced the same kind of selection pressure and may or may not have answered it in the same way. And then I'll talk at the end briefly about our current stuff on paralogs. So this turns out to be a gene family that's involved. Right? So here's the natural history below the organism. Right? So this is the sodium channel that you have in your nerves and muscle, more or less. Right? So again, not a neurobiologist, not a biochemist. The sodium channel is membrane bound. It's got 4 homologous regions here, color-coded. They're named 1, 2, 3, and 4. And they have this thing called a P-loop, which goes in and out of the membrane, which is where TTX binds, as it turns out, but also where the gate for sodium is. So these are voltage-gated channels. Sodium comes through here, the gate opens, it moves down, and that's how you get this rapid depolarization of the membrane. right? So what we know from old work on sodium channel structure is that TTX binds to the P loop in domain 1. And I won't bore you with all of the data, but we see nothing going on in D1 or D2. So there are no changes of interest there. Evolution has pushed snakes to action here in D4, and I'll show you those data. So before we got to the genes, we had to figure out what the mechanism was. And we recruited a biophysicist named Shanna Geffney, who's a postdoc at Stanford right now. And she did a really old school textbook prep that I still love because her advisor at the time told her, don't waste your time, it's not publishable. And she did it anyway to humor us, and she got a science paper out of it. So don't listen to your advisors too seriously. All right. So she took muscle out of a snake, she pinned it down in a saline bath, and did patch clamps to record the voltage propagation, and then added concentrations of TTX. And so if you do that to a mammal muscle, it takes very little TTX to completely arrest channels, and you would move from this nice black control action potential to a flat line. When she does it to snake muscle, here are two representative snakes, one from the Oregon hotspot and one from the California hotspot. You see that action potentials are slowed down at a dose that would completely arrest mammal channels, or unaffected, depending on the snake. Right? So there are clear differences in the membrane function that suggest that it has to do with sodium channel structure or sodium channel qualities that confer these population differences in resistance. She did the same sort of thing for individual snakes and showed that there was individual variation within each of these populations that corresponded to organismal level resistance. Right? So then she took the molecular approach and said, we don't have a genome for snakes. We don't have any idea what the sequence for snake sodium channels is. So we're dealing with the sodium channel in skeletal muscle. So she took a human primer and tried it, and it worked. So this is dumb luck, right? She was able to get out the coding sequence for all 4 of the domain P loops, look at all the substitutions and compare them to a baseline. So this is a non-resistant snake. This is the amino acid sequence for domain 4. That's the same sequence that you have or a mouse has or a chicken has. It's incredibly conserved across vertebrates. She does this for a resistant snake. She finds 1 substitution. She does it for another population of snakes, finds a couple of substitutions, one of them in common. And then for this California lineage, remember this is a different clade, so it doesn't look like that here, but this is probably a second origin of resistance. She gets 4 substitutions. Again, this one valine is in common. So I look at this as a naive natural historian, I guess, and thought, well, that's obviously the most important change because it's present, probably arose twice, and it's present in every lineage. And she says, well, you can't tell anything about the function from this. All we have is a correlation that says there are changes that correlate with population level. So she then takes the gene and does site-directed mutagenesis and puts that into a xenopus oocyte and does membrane readings on that oocyte. So she's expressing the gene in a different genetic background to ask what does that protein do itself when it's challenged with TTX. So real functional work. And she did 2 things. She took the whole snake sequence, so the whole coding sequence of that gene, more than just the P loops, but all 4 domains. And then for each of these, she made a chimera based on human sequence, where all she did was substitute these amino acids that are implicated in our phylogeny. Right? So a real test of whether or not it's those amino acids or some other part of the gene we're not focused on. And in a nutshell, she gets basically the same pattern of resistance that we expected. Non-resistant, there should be a green line here for the weakly resistant population, the Benton County, and then the Willow Creek. The open symbols are these chimeras. You can see that the chimeras are maybe a little more resistant, but not much different. Probably within the scope of the technique, they're no different than the whole snake sequence. So that really nails down that it's the amino acids that we had in mind that were responsible. Out here in red, these are 2 chimeras, and this is another one of those dumb luck stories. She forgot in the round symbols, the circles, to include this valine. So she had a mis-experimental design. But it turned out to be fortuitous because this was the one that we thought was really important in conferring resistance. And you can see that there's maybe a twofold difference in resistance. So this thing that looks phylogenetically like it's the most important is not the most phenotypically important. In fact, it has a relatively weak effect on the resistance phenotype. And in retrospect, that makes a lot of sense because if this yellow population or green population from Warrenton had it and it was a tenfold effect, you'd expect a much bigger jump. So we see that the most common one has a small effect, but also that this N right here has a tenfold effect. And that's probably the answer for why some populations get away. It turns out that that is a single base pair change to a single amino acid change that has a tenfold effect, an order of magnitude effect on phenotype. So I don't like the idea of a saltational step in evolution, but that's what it looks like. Right? So we've got one big jump in phenotype that essentially removes them from that ecological interaction. Right? So now we're looking at multiple species. Can we tell anything about the geography of the interaction from looking at multiple taxa that seem to be involved? So for a long time, all we knew about were Sertalis eating newts. But once you start being known as the newt snake person, you get all these emails of, I saw this thing eat a newt. Somebody found a dead heron, and there was a newt in the belly. Could that kill it? Sure, yeah. I have more emailed pictures of dead things with newts in them than you can imagine. All right, so what was beautiful about this was people out in the field doing natural history observations led us to start looking at Atratus, the coastal aquatic garter snake, especially around here in the Molino Creek area just south of the San Francisco Peninsula, and Cauchy over here in the southern Sierra where they had seen them eat newts. So these are Turosa over here and Granulosa or Turosa here, in places that we had never been. And as we fill this out, it turns out that we have as much variation in resistance within Cauchy and within Atratus as we do within Sertalis, and I'm not going to go into that. But the patterns of resistance were the thing we initially were interested in, trying to figure out if you got a common pattern in these overlapping ranges in the two different predator species. All I can really tell you is the answer is no, and I don't have an answer to why it's no or what structures the pattern. So Chris Feldman took these data and said, well, these are really an interesting way to ask about the standing variation question. Right? So if there are alleles out there in the family, the subfamily Natricines, then we ought to see a pattern of similarity, a topology of the gene tree that shows all of those resistance alleles in sodium channel 1,4 as close relatives. If these are new mutations on the way to evolution in each of these lineages, then we should see a topology that maps on more closely to the species tree, right, as shown above. So here are the data that he got. And you probably don't even have to get to the phylogeny to see that these are new mutations. So here are a bunch of different Thamnophis and a few other Natricines as outgroups. Their resistance levels. Here's the California Sertalis that I showed you before. And what you see with Atratus is, first off, not only do we see not the same changes as Sertalis, but we see changes in domain 3, so that's this region here, that we never see in Sertalis. So we've got a whole other piece of the gene in play when we go to this other species. There are some of the changes in common, so that N that's so important phenotypically is present in Atratus on the coast. But these new mutations we've never functionally expressed, and we don't actually know what they do. We just assume that they're important. Cauchy has none of the changes in domain 4 that are present in the other two Thamnophis, has one new position just one spot off of one of the Atratus positions. So these are, we know from reconstructions, in the region that should influence binding to TTX, but we don't know functionally at the same level that we do for Sertalis whether they really come into play. Right? If you match up the topologies, they match up really nicely. Right? So this looks like the Sertalis allele has arisen along the way to Sertalisdom, and the alleles in Atratus and Cauchy likewise in that lineage. So no evidence that we had standing variation at all. The other thing that we've been interested in is whether we get introgression. It's not infrequent hybridization among garter snakes, whether we could be seeing movement of alleles back and forth across these species boundaries. And there doesn't seem to be much evidence of that. Really clear when you find different alleles in different systems. Right? So then to go a step further out, Chris, this is another one of these don't do this at home grad students. Chris said, well, why don't we take all the snakes in the world, not literally, but take as many snakes as we could get in the world and look at their sodium channel genes with the expectation that there are some species that eat amphibians that have TTX, and maybe they show the same patterns. And we said, that's a lot of money and a waste of time. So Chris did it anyway, and lucky for us, right? So here's the data from the last slide. Sertalis couchiatratus, these are the changes I just showed you. Here's the reconstruction of the sodium channel. This is amphisma from southern Japan. It eats a newt named Cynops that has TTX. This is a Southeastern Asian keelback rhabdophis that eats Polypodates that has tetrodotoxin. In South America, we have a xenodontine Lyophis that at least used to eat Atelopus. There may not be any Atelopus anymore, but they were known to have TTX. So we've got 3 candidates here, and actually one other I'll hope to remember to mention, that all eat TTX-bearing prey. When he ran the sequences, and he's done this for 1, 2, 3, and 4, there are no changes in 1 and 2, he finds that amphisma has exactly the same substitution as Atratus in domain 3. Rhabdophis has a change 1 off of one of the key Sertalis substitutions. And Lyophis has the same 2 positional substitutions as Sertalis, right in that core that had that 10-fold effect in resistance. Right? So dramatic levels of constraint or convergence in driving the evolution of the sodium channel gene. Right? So here is the tree that he was playing with. He sampled every one of those taxa. So this isn't cherry picking and getting lucky in that sense, but really, here's Lyophis, Rhabdophis, amphisma, really taking a wide cut and finding only these species that we think eat tetrodotoxic prey showing those changes. So this is a gratuitous membrane, gratuitous channel reconstruction, just to show you where they sit with tetrodotoxin in the middle. All of them line up in the pore with the exception of this one. That's the one in domain 4 that sits on the outside, and it's not clear what that does. It's not been expressed. So Chris had this idea that one of the reasons we see such high constraint is that there are costs to these mutations. And so he dug into the site-directed mutagenesis literature. So most of this is done in mouse. These are not the substitutions from snakes. But if you do a reading of channel conductance and its block on tetrodotoxin binding, you see a really strong negative relationship. The mutations that have the least effect on TTX have the least negative effect on conductance. The ones that are most dramatically affecting the blockage, the binding, are the ones that really hammer the function of the channel. And the only one of the snake channels that we know, snake substitutions, that is in common with this data set is up here at the top. So the expectation is what evolution's doing is picking a poor way to accomplish this job by not sacrificing the cost or not incurring so much of a cost. So the last slide, last piece of the puzzle, that arrow was to remind me to mention the story is not quite right. Oh, those moved. That should be down here, Heterodon, and this is actually the mountain lake Sertalis. There are two species that we've taken sequence on that eat tetrodotoxic prey that have none of these substitutions. One is the hognose snake in the eastern US that eats Nodophthalmus, and the other is Virginia Thamnophis sertalis that eat Nodophthalmus. Both are resistant at the organismal level. Neither has any recognizable changes in NAV14. We have no idea what's going on. Right? So this is the last piece. What we're currently doing is trying to explore the paralogs of sodium channels. So these are a family of anciently duplicated genes in vertebrates. We have dealt with 1,4 expressed in skeletal muscle in mammals. There are some channels like 1,5, 8, and 9 that are natively insensitive, so they're not bound by TTX. So if you were to be injected straight into the heart with TTX, you would be okay, as long as the blood didn't move it out. Guess that can't work. So what we're trying to do is even find out how many of these genes occur in snakes. It looks like probably the full complement. And then ask whether you see the same sorts of changes. This is a highly conserved family of genes in these other tissues. So up to date, all we've been able to do is get the sequence on NAV17, which is a peripheral nerve channel. Right? So as a reminder, here's a human sequence, Atratus sertalis for the 1,4 gene. In NAV17 in sertalis, you see one of the same changes as you do in 1,4 in Atratus, and some of the same changes that you see in 1,4 in sertalis. So not only are they the same changes in the same species, so we were thinking about recombination as a way that you could move some of these adaptive changes across genes within a family. Some of them are found on the same chromosome. But you've got other changes that are clearly not represented in the other genes within the family. So this is really preliminary. We don't have a whole lot of this picture. But in getting into this, it surprised me that there are not a lot of predictions about convergence within gene families within species. So it's the same sort of argument that we're used to dealing with across taxa. Right. So that's where we are now. This is really the big pulse. And eventually what we hope to do is be able to develop some population genetic tools to go back out into these populations and look at selection, changes in frequencies in these populations where we think selection's strong and where we think selection's weak. Right. So what did I just babble on about? Messages to take home. Geographic structure is critical, both in structuring the dynamic but also in being able to reveal what the dynamic is like. So our ability to tell what's going on in an evolutionary arms race is difficult if you just have one point in time and space. So we're essentially using populations or localities as a surrogate for time. And there are better ways to do it than we've done, where you could actually look at time as one of the dimensions. And we hope to be able to do that later on. What we think are evolutionarily intense hotspots based on phenotypes might not be current hotspots. These may not be that critical. We know that functional changes in the genes that structure the proteins are important in this system. This has been a hot topic lately, obviously, where people are really focusing on the role of expression differences. This may be a result that's as much a function of the kind of adaptation we're looking at, where having more of something probably doesn't have much of an effect. If the toxin is so powerful that it saturates all the available channels, expression differences may not be an option. In our case, it looks like these are new mutations, and convergence among these lineages really paints a strong picture of constraint. Again, this may be a function of the kind of thing we're looking at where sodium channels are enormously conserved. I mean, you really see almost no changes in that coding sequence in the P loops across the family of genes within a species or across all of vertebrates. So these may not be very general explanation or very general conclusions. They may be really particular to the kind of adaptation that we're dealing with. All right, so I get the fun of telling good stories. These are the people who do the work. Charles was the HPLC Newt person. He's the only person in North America who's ever made the HPLC work for tetrodotoxin. It takes a technological skill to do that. Shanna's the person who did all the nerve and the early molecular stuff. Joel McLaughlin and John Chukalowczyk are doing that molecular work on paralogs right now. Chris Feldman did all that wide phylogeny comparison stuff. So many other people have helped us. We've been very lucky to have a lot of undergraduates help us in the field. I was once told in a grant review that Brodie is little more than an overpriced snake collector. It was the nicest thing anybody ever said about me. So thanks for listening, and I'll enjoy my cookie.
So why don't those of you who need to go feel free, and then I'll let Butch take about 10 minutes worth of questions, and then it's time.
time to refresh them, as we say.
I need refreshment.
Questions?
Disperse.
Have you looked at any effects of predation on the snakes as a result of newts? Do they benefit at all from any newts?
It's a beautiful question.
Could you repeat it? Would you repeat it?
Yeah, sorry. Have we looked at predation on snakes? Is there a benefit to snakes of eating newts? Yeah, it's a long answer because I'm obliged by a pact within the lab to explain this story. It's known as the Susan hypothesis, after my wife, who came up with the idea and was poo-pooed for 4 or 5 years until we started to take it seriously, and I was shamed into agreeing to always introduce it that way. Fairly so, I might say. What you might have noticed is that some of the snakes have these bright red heads and red patches on the side. Those are the snakes that are most commonly, those are the snakes that are in the populations with the highest level of toxicity of newts. So another bit of work that Becky Williams has done is to look at the retention of toxin in the snake. And the toxin sticks around for at least 7 or 8 weeks after they've eaten a newt. So there's the potential that I don't want to call it sequestering, but that toxin sticks around and is an anti-predator benefit. And the Susan hypothesis specifically is that they've evolved red coloration as a warning signal, which I thought was crazy and maybe isn't so crazy. The problem with the whole story is that the toxin in the snakes is concentrated in the liver, which is not a great place to defend yourself, you would think. But there are also these natural history observations from the snake dens in Manitoba, which are not, they don't eat newts. Crows come into the dens and flip snakes over and eat a piece of the liver out. The liver is the only organ that apparently regenerates, and the snakes survive. And you can go to those populations and see which snakes have been attacked by the scars right over the liver. So it's a long answer. So yes, I think there are potentially advantages to the snake in terms of what they acquire from the prey. Sorry that was so long.
Do the crows sample the liver and then make a decision whether to eat based on the? I mean, that's just like a three-way.
So that's in a place where where there are no toxic newts.
Why are they eating?
Why are they sampling the liver?
Because it's full of toxins and crows are adventurous.
So they get-
I don't know. I have no idea. I have no idea why they do that.
Who eats crow? Sorry.
Yeah. I have. Yeah.
In one of your slides, the one where you had to wave your hands, you spoke about a region of phenotypic space where there's conceivably a possibility of an arms race going on.
Yeah.
My question is it really likely, or do you have evidence that in fact the garter snakes are the main selective pressure on
Yeah.
Tarika with respect to their TTTS?
I think it's. I don't think it's. I don't think that there's a good counter for other selective pressures on tetrodotoxin toxicity. So there isn't really anything else on the adult stage of the newt that preys on the adult stage of the newt that's able to ingest that much toxin. So what Brian Gall is angling on now is that newts put tetrodotoxin into the egg. They apparently defend the eggs with TTX. The level of toxicity in an egg is closely correlated to the level of toxicity of a female. So it's essentially a correlated phenotype. And caddisflies are major predators on amphibian eggs. Western caddisflies, he's just shown, are resistant to TTX and avoid highly toxic eggs. So there's some potential that there's predation at that life history stage that at least helps along that interaction. So is that what you're thinking
Yeah,
of?
I just wondered how much is just a arms race between those two
Yeah.
players versus, as you mentioned, the
Right,
caddisflies
yeah.
are
So I think
complicated.
it's completely reasonable at other life history stages that there are interactions like that. But at the adult stage, there's really no other opportunity. And I should be fair and say, we talk about arms races, but we have done very little to show the other side. There's very little evidence that the newts are really keeping pace, and we don't even know that the newts make the toxin themselves. So it could be bacteria in newts.
Do you have a sense of what the population densities of the newts are across the range?
No, not really. Yeah, no. I mean, the answer to that's I wish. They are the most obvious amphibian most places that you encounter them. But that's obvious to a human. And they come into these big breeding aggregations in the early spring. So they're easily encountered. The data that we've always wanted to get are relative prey abundances. And our biggest problem is you can't be in multiple places at the same time. So yeah, nope, I got to bow out on that. Brett.
So what you showed in your maps that there was an area of high toxicity and high resistance, if I've got it right, sort of in the Bay Area.
Yeah.
And then also across the Central Valley over in the adjacent Sierra foothills. And you said those were probably independent, phylogenetically sort of independent high levels, right? But as I'm sure you know, there's a whole bunch of species of amphibians and reptiles that do this Trans-Valley leak, where they all move right across right exactly that area. And so I guess what I'm wondering is that it just seems like an amazing coincidence that they would line up twice and it wouldn't be that the genes moved across, perhaps as part of a hybridization event or something, but you know, I mean, everything does that. They all go across there. And so what do you, I mean, how strong is the evidence that those are independent and it's just an amazing coincidence?
Not super strong, but the evidence is in the structure of the alleles. So phylogeographically, there's pretty good support that the Sirtalis clade over there in the Sierra is independent of the coast clade, that it's pretty well separated. But that's a little bit squishy. The allele, so the Cauchy over there are very resistant. They have a different allele than the Atreides, different allele than the Sirtalis. The Sirtalis populations over there in Placer County don't have the same allele as, they have one amino acid different structure than the ones in East Bay. Doesn't mean it wasn't a gene that moved over and lost or gained an amino acid, but it's different, and that's what we're basing it on. The other thing that's weird about that part of the map is that elegans, which is another garter snake, that sometimes eats amphibians, but isn't really a major predator on, doesn't seem to be a predator on Tarica, only in that area has some resistant individuals and some resistant alleles. And that's why we think hybridization is going on there. So it's squishy for sure.
And on the newt,
Yeah.
the newts over there are different species than the newts on the east side, on the west side, right? So on the newt side, could that be, you know, a sort of hybridization?
That could be, that could easily be, if it's a genetically based toxin, moving the toxin across. We have I wouldn't say much about the toxicity being independent over there. Yeah, although if you go to the northern Sierra, they're not toxic up there.
Let's do one more.
Okay. Scott.
The degree of diet specialization in the predator should strongly influence the probability of the coevolution that you're probing. Are there differences that you know of globally in the degree of newt specialization in the different snake taxa that you've been examining?
No, not that I know of. I'm not sure those data are known. Those other snakes are really poorly known. You know, getting a tissue from one is tough. So getting diet data of any meaningful level is tough. So I agree, but I don't think there are any data yet.
What's your intuition as the person whose intuition?
My intuition is, I'd have to ask my dad because he's always right.
That's your wife.
God, they're all right. They're always right. My intuition is that it's not different. That these are rare events, and that part of what drives this is the severe cost of an accidental interaction with a toxic prey. So that it has less to do with exploiting a rare exploitable food source, and more to do with a severe consequence of encountering something accidentally that's going to kill you. So, all right.
Which proves that snakes are dumb. Because
Snakes have very small brains. So,
but not bush. Thanks
yeah.
very much.
Thanks, guys.